Okay, this is not the end of chapter 7, but it's the end of chapter 7 for the third exam. So this is the last video, and it'll be the last one in the file for the third exam. We'll pick this up after the third exam with another video and another file. We're going to look at mutations, where it's an embarrassing moment at Gene parties. My goodness, Vinny's mutating. These guys are supposed to be chromosomes. Right? He's a messed up chromosome. And then, warning, underground nuclear test area, and you're seeing, a lot of mutated groundhogs. Radiation does indeed cause mutations. Any change in DNA from what it's supposed to be to something different is a mutation. So it could be a really tiny change, it could be a really huge change, and everything in between. They're all mutations. We're going to start with the little ones. So you make a copy, and you wind up swapping out one nucleotide for one that's not supposed to be there. You substitute one nucleotide for another. We talked about this when we had the table up. A lot of times, you're changing a codon, you're technically producing a new allele. Anytime you change the code, it's a new allele, even if it doesn't really do anything. And a lot of times when you change a nucleotide, it doesn't do anything. You get the same amino acid you would have gotten anyway. That's called genetic redundancy. The system is redundant, where you can change stuff and it doesn't really do anything. Extra stuff doesn't get you anything different. So we swap out an A for a G, and it changes the glycine we would have coded for serine, which are very, very similar small amino acids, probably does absolutely nothing to the actual coiling and three-dimensionality of the protein. But a lot of times you could swap it out and get glycine again. This is a deletion, where that U doesn't get put in. You wind up skipping over one of the nucleotides. This causes what's called a reading shift, where you not only change that codon, but you change everything downstream, because what you've done is you've taken that codon and you bumped it up one. And that codon, you bumped it up one. That codon, you bumped it up one. Imagine a book. You get a page in a book, and the computer that's doing the printing has a little glitch, and it swaps out one letter for another. Book's still going to be pretty readable. Probably, you'll go, you see one word, you may even not notice that it's not spelled right. But say the glitch in the computer takes every space between every word and moves it one letter in either direction, so that every word becomes either one letter longer, it picks up the first letter of the next word, or one letter shorter, it picks up, it pushes its last letter onto the next word. It's unreadable, because none of the words are going to make sense, and especially not going to match what the book was originally supposed to be. So these, either an extra one goes in and bumps everything one down, or one comes out and moves everything one up. If you shift the reading list, sorry, I said it, now it's not in my head anymore. The reading frame, you shift the reading frame, then you're going to change this. It does turn out that if the messenger RNA is going to be spliced, that this reading shift will only go to the next splice point, and then the next spliced area, the next exon, is going to be okay. So that's another reason why introns and exons kind of are useful, because they can minimize the impact of some of these. But these tend to be big deal mutations. Anything that changes the whole sequence or most of the sequence of the amino acids is going to give you a very different protein than you were supposed to get. There's some mutations that we talked about, DNA parasites. Little bits of DNA that wind up inserting themselves into DNA elsewhere. They're often codons, little clump of 3 nucleotides that wind up in the middle of a gene. What does that do? Well, it does change the reading frame, sort of, but instead of changing all the codons, what it does is it puts an extra codon, puts an extra amino acid into the protein. And that may or may not have a big effect on the protein. Huntington's disease does that. But what they found is that those little, what they call a triple repeat parasite, will duplicate itself and make that little, it starts out as a triple repeat in the gene, and then it becomes 2 of them, and 3 of them, and 4 of them. And every time it does that, whatever amino acid that's coding for winds up in the strand of the protein, and it makes the protein longer and longer, and maybe it doesn't hit the right curve point, or doesn't curl over properly. And so the longer that parasite grows, generation after generation, the worse Huntington's disease gets. So somebody with Huntington's disease is likely to have it worse than their parents and worse than their grandparents. For a long time, that was a big mystery. Some mutations involve pieces of DNA. You can pick them up if you get multiple breaks in your DNA. The proofreading machinery just reads broken ends. It doesn't know where they came from. Under most conditions, there's 2 broken ends, they put them back together, because most of the time your DNA just breaks in one spot. If you get multiple breaks though, it just starts piecing ends together, and it can take a piece that was in one place and put it someplace else in your overall DNA. It may move it from one chromosome to another, it may move it from one spot on a chromosome to another spot. It's a translocation. And if there are active genes on that piece, then it could wind up going from a part of the nucleus that's easy to get to, so you can really easily use that gene, to one that's hard to get to. So that gene winds up not being expressed as often. Or the opposite, it may wind up being expressed more often. Is some people think that the reason our brains are a little bit better, I don't know if that's true, than chimps, is that some of our brain proteins are in different spots on the chromosomes than theirs. And they think there might be a position effect that makes us use that better. I'm not really sure, but it's suggested. In some cases, you get what are called inversions, where a piece gets put back in, but it's put back in the wrong way around, in which case it'll get read weird when you go, if there's a gene on that stretch that you need to find. Some things generate mutations. They're called mutagens. There are chemicals that do that, and radiation does that. In radiation, certain types of hard radiation shakes your DNA and causes it to break in very often multiple places. Some chemicals do the same thing. And then your cell is scrambling to put everything back, but it's not necessarily going to put it back right. In some cases, with a heavy radiation can actually screw up your proteins too. That's really bad. But mutational radiation, you may wind up with a piece that instead of going back into a chromosome, all the pieces are put back and there's this extra piece wandering around and there's no place to stick it. There's no other broken ends. So you wind up with a piece that if the cell divides, it makes a copy of it. But part of your centromeres connect to the cables that pull the chromosomes apart. This thing doesn't have a centromere, it's just loose. And it's wandering around inside your cell once the nucleus goes away during division. And there's a very good chance when your cell divides, you've made copies, both of them wind up in the same cell. So one of the copies doesn't go that way and two copies goes the other way. And if there are important genes on there, it could really mess up the cells that you're making. That's one of the reasons why we use hefty doses of radiation to try to kill cancer cells, because they're dividing and dividing. We're trying to break their DNA up like crazy so that when the cells divide, they get extra pieces and missing pieces that can potentially screw them up. Radiation is not going to damage cells that are not dividing typically, because the chances of you hitting important breaks right in the middle of a gene that that particular cell is using are pretty astronomically rare. But losing a piece off that's got working genes in it is a little bit more common. So we hope that we hit the cancer cells, we hit them with a bunch of beams all at once, really dose the hell out of them, with beams that hopefully are not causing too much problems on the way in and way out. That's why we overlap the beams in one spot where your tumor is. And hope that we break up the DNA so much that most of the cells divide and then they're genetically screwed up and die. We have chemicals that do that too. There's a term called aneuploidy. I'm not going to put that up there. But sometimes we get a division mistake where there's an extra chromosome or a missing chromosome. If you are conceived with an extra or missing chromosome, chances are having an extra copy of all those genes or a missing copy of all those genes on that particular chromosome is going to screw up the chemistry of your cells early on, and you will not survive very long. Is that Down syndrome is about the only survivable chromosome extra mutation that you can be conceived with. And it still messes with your development, right? It's not without effects. And in fact, most Down syndrome fetuses don't make it to term. But all the other chromosomes, except for the X and the Y, if you get an extra one when you're conceived, chances are we'll never know you were there. Maybe you'll show up on a pregnancy test really early. But it's gonna come back and kill you pretty quickly. And number 19, occasionally fetuses survive to term, but they're very messed up. But the other chromosomes, really big, lots of genes there, extras, missing, bad. I mentioned before, as women get older, they have a greater tendency to have division issues, making egg cells. And that's why Down syndrome is more common in older women. And at least sort of, women have more difficulty getting pregnant. What really happens is they get pregnant and that sperm and egg get together, but because of extra and missing chromosomes and all the other chromosomes being an issue, if you get an extra and missing one, the embryo doesn't live very long. So they don't really know that they were ever, fertilized, that there was actual conception that happened. Okay, that's the wrap-up point for this particular part of going into the third exam. And then we'll pick up this chapter with cell division after the third exam.